Hello, everybody. So we're going to be here today going over our fundamentals of the nervous system and nervous tissue lecture. Chapter 11. So the nervous system, its job, its function is to control and integrate all the body activities that help maintain life. There are three overlapping functional systems here. There is the sensory input, which is basically taking information from a sensory receptor, monitoring some sort of internal external change, and then sends the signal towards the brain. Within the brain, there's the central nervous system, there's the integration process where we are processing and interpreting this input information. And then we send out a signal to the motor system, motor output, basically responding to that change. So in this example here, sensory input of the eyes sees the water, integrates, realizes within oneself that I am thirsty, sends a signal out from the motor output to basically pick up the water and drink the glass. So the nervous system itself is broken down into different areas. Two big main ones here are the central nervous system and the peripheral nervous system. So the central nervous system is consisting of the brain and the spinal cord. It's abbreviated to be the CNS. So the CNS occupies the dorsal body cavity. It interprets sensory information and dictates motor output based on reflexes, current conditions, and past experiences. The peripheral nervous system, also known as the PNS, is basically consisting of the spinal nerves, the cranial nerves, and the ganglia, which are collections of neurons and cell bodies. So you can see here, the CNS is the brain and the spinal cord, and then the peripheral nervous system are the branch outs that are close to that area, such as the cranial nerves, spinal nerves, and the ganglia. The peripheral nervous system is outside of the central nervous system, consisting of the nerves that extend from the brain and spinal cord, as well as the ganglia. The spinal nerves basically carry impulses to and from the spinal cord, and cranial nerves carry the impulses to and from the brain. And this relationship can be broken down further. So here is a graph looking at basically the central nervous system and the peripheral nervous system being the sort of main jut outs, of which you can see that they communicate with each other back and forth. The central nervous system being the brain and the spinal cord, working on integrative and control centers, whereas the peripheral nervous system is the cranial nerves and spinal nerves, and it's the communication line between the CNS and the rest of the body. Now, the peripheral nervous system can be broken down further. We have our sensory or afferent division. So this is the sensory, somatic, and visceral sensory nerve fibers that take in the stimuli and information from our environment, and they conduct these impulses towards the CNS. So they use the peripheral nervous system to then send it over to the central nervous system. And then we're integrating and we're sending back out through the motor efferent pathway. So this motor efferent pathway is signaled from the central nervous system out towards the rest of the body. This is consisting of the motor nerve fibers, and it conducts impulses from the CNS to the effectors, such as muscles and glands. The motor division can be broken up further into the somatic and autonomic nervous system. The somatic nervous system is our voluntary movement. So it conducts impulses from the CNS to the skeletal muscles for voluntary controlled movement. The autonomic nervous system is more about those automatic sort of stimulatory processes or movements. So visceral motor pathways that are involuntary, such as the heart, the internal organs, smooth muscle, digestion things, and certain glands. So this is more involuntary, this autonomic. And this autonomic nervous system can be broken up further into sympathetic and parasympathetic. Sympathetic, the best way you could think of this is fight or flight. This is the mobilization of your body systems when you need to increase your stimulation, to increase your blood flow, increase the release of adrenaline, increase your heart rate, increase your ability to respond to some sort of threat, right? Or maybe we're just nervous, but for some reason, these body systems are speeding up. Conversely, the parasympathetic is known as the rest and digest. So this is calming down your body, conserving energy, and it promotes housekeeping functions during rest. So we'll talk about these a little bit more here. So the functional divisions of the peripheral nervous system, specifically the sensory afferent and the motor efferent. These sensory afferent pathways transmit signals towards the central nervous system, whereas the motor or efferent pathways will transmit signals away from the central nervous system. And there's two main motor divisions. There's the somatic nervous system for voluntary skeletal muscle contraction, and the autonomic nervous system for involuntary cardiac muscle, smooth muscle, and other glands. The nervous system is basically composed of two main types of cells. We have neurons that make up all the sort of nerve body, the main sort of nerve cells, and then the neuroglia, which are basically nerve glue. So the neuroglia are these small cells that are associated with larger neurons, and their functions are supportive in nature. So they support scaffolding for the neurons. They produce different neurochemicals, and they help insulate the neurons. And again, the nervous system is mostly made up of nervous tissue that is highly cellular. So these neurons, they are about 80% cell space. Only about 20% of the space is extracellular. And the neuroglia, while less commonly talked about, make up the majority of your cells. So backing up here really quick. You have different neuroglia cells, whether you're in your central nervous system or peripheral nervous system. So we're basically going to walk through the different cells within the different systems. So first, we're going to talk about the neuroglia of the central nervous system, of which we have astrocytes, microglia, ependymal cells, and oligodendrocytes. And again, these four will be associated with the central nervous system. This astrocyte, its main function, it's a star-shaped, most abundant type of neuroglia that holds the neurons in place. They help link the neuron with supporting capillaries within the region. And they also help mop up any potassium ions that need to be recaptured and recycled. The microglial cells, they have these thorny processes that monitor the health of different neurons. And then if they find something that shouldn't be in the area due to injury or different debris, they'll basically get rid of it through phagocytation. This is a very important role because there's not a lot of immune system stimulation within the central nervous system. So these microglial cells help clean up things that should not be there. We have ependymal cells, which are basically a wrapping or garment. This helps line the cavities of the brain and spinal cord. A lot of these are facilitated through cilia-based movement. And what they do is they help move the cerebrospinal fluid within the cavities of the central nervous system. So that's the function of ependymal cells. Then we also have oligodendrocytes. Their main thing is wrapping the central nervous system fibers in what's called a myelin sheath. This myelin sheath basically helps more quickly conduct the nerve impulses of the central nervous system or the nervous system in general. So myelin sheaths help with nerve impulse conduction. And the central nervous system myelin sheath cell is the oligodendrocyte. So we've also got neuroglia within our peripheral nervous system, of which there are satellite cells and Schwann cells. Satellite cells function similar to the astrocytes of the central nervous system, where they are surrounding the neurons in the different cell bodies, and they help support, brace, and communicate between the neuron and the nearby capillaries. Schwann cells are similar to those oligodendrocytes of the central nervous system, where they surround and form myelin sheaths around the peripheral nerve fibers, which again, the whole point of that is to help conduct the nerve impulse. Neurons are basically nerve cells, which are the structural units of the nervous system. They receive and transmit messages in the form of nerve impulses, and they have three characteristics. They have an extreme longevity, so you keep your neurons for most of your life. They do not have a high turnover rate like many of your other cells. Somewhat associated with that, they are amitotic, which basically means that they lose their ability to divide and reproduce because they're not being turned over, they're not needing to be renewed, so they're not renewing themselves because they last your whole life. And they have an extremely high metabolic rate. They need an abundant supply of oxygen and glucose. Glucose is the main fuel source for your central nervous system, so you need carbohydrates and glucose to be able to basically think and conduct nerve impulses. And it uses a high amount. About 25% of your daily calorie intake goes towards supplying your neurons. And there are specific structures associated with the neuron. So first of all, I'm going to look at the second one here, the cell body. The cell body is the biosynthetic control center. It has a spherical nucleus, and it's essentially this whole big body portion here. The axon you can find starting down here and branching down. The axon transmits signals away from the cell body. And then the dendrites you can see up here. The dendrites receive the signals that enter the cell body. And clusters of cell bodies within the central nervous system are considered nuclei, and clusters of cell bodies within the peripheral nervous system are considered ganglia. You'll also see a lot of structures similar to a rough endoplasmic reticulum for protein synthesis, basically synthesizing a substance called chromatophilic, which you can see right here. Most of our cell bodies are located within the central nervous system, where they will be protected by the bones of the skull as well as the vertebral column. So when we look at how do we classify neurons, basically there's always a functional and a structural, right? So neurons are no different. Neurons are classified by their structure and their function. From a functional standpoint, are they sensory afferent type neurons? Are they motor efferent neurons? Or are they interneurons? Interneurons connect sensory neurons to motor neurons, and they make up about 99% of your neurons, as well as the structural classification, multipolar, bipolar, or unipolar. So do they have, it's all about how many branches off there are from that main cell body. If there's 3 or more, it's multipolar. 2, bipolar. 1, unipolar. And of course, we'll look at some examples. So these multipolar type of cells are going to be the most abundant type of neuron within the body. they have several dendrites in a single axon. And they also, so the type that you'll see utilizing this multipolar configuration are generally motor neurons and interneurons. The bipolar configuration is single dendrite and single axon. You'll see these in the sensory neurons for special organs such as the eye or nose. And then unipolar are single axon. Generally, the rest of the sensory neurons function in this way. So, of course, we're going to look at some pictures. So basically, it's how many projections are there off of this cell body. With this multipolar, there are many different dendrite portions as well as an axon portion. With the bipolar, see the cell body, and then basically two branches. So one branch here, one branch here. So that's a bipolar configuration. Unipolar is the cell bodies right here, and there's only one branch off, and this is the axon terminal. So it's a single branch off. So that's really just the main difference between the structural classifications. How many branches off are there from the cell body? And of course, they're associated with different types of nerves within the body at different locations. So make sure you know that. And then from a functional standpoint, is it sensory, motor, or interneuron? So our sensory or afferent pathway, these are going to transmit impulses from sensory receptors in the skin or internal organs towards the central nervous system. Generally will be unipolar. Whereas the motor efferent pathways transmit impulses away from the central nervous system towards the effector organs, such as muscles and glands. These motor neurons are typically multipolar. And then interneurons lie between the motor and sensory neurons where the integration process occurs. These are generally multipolar neurons as well. Oh. Did I miss one? No, we're good. So another thing, so we talked about how there are specific types of cells, specifically the Schwann cells and the oligodendrocytes that have this myelin sheath associated with them. Well, let's talk about what that does. Excuse me. The myelin sheath essentially helps protect and electrically insulate the axon. So this helps increase the speed of the nerve impulse. So a myelinated neuron will conduct an impulse faster than an unmyelinated. In the peripheral nervous system, this is done by Schwann cells. And in the central nervous system, this is done by oligodendrocytes. And then between the myelin sheaths on the axon, so there will be space in between individual sheaths. You don't really see it here, but imagine there's two of these sheaths. There's space in between them. The space in between is this gap called the nodes of Ranvier. So these are myelin sheath gaps along the axis, and the impulse has to jump from node to node between the sheaths. So within the central nervous system, the oligodendrocytes form the myelin sheaths. So within the white matter of the brain, you'll see regions of the brain and spinal cord that contain dense collections of myelinated fibers. And within the gray matter of the brain, you'll see regions of the central nervous system containing cells that are unmyelinated. So there's going to be a difference in the conduction speed of the impulses from that myelination. And again, within the central nervous system, this is dictated through the oligodendrocytes. If it's the peripheral nervous system, it's the Schwann cells. So now we're going to basically talk through some basic principles of electricity, and then we're going to talk about how that's related to how nerve impulses conduct their electrical signaling. So the human body is full of a bunch of different substances that have different charges. Some of them are positive, some of them are negative, some of them are neutral. But when you sum total everything together within the human body, it comes out as being electrically neutral. So it has the same number of positive ions and negative ions within it. But if you look at a localized area, like this is just a random example, but a left arm, like you may see concentrations that are more positive or more negative. If you look in one specific part of the heart, you might find a place that's more positive than negative. But when you sum total the whole body, we're neutral. And basically, opposites attract. So since positive and negative attract towards each other, you have to use energy to separate them. And generally, they're going to be separated by some sort of membrane barrier, where the configuration ends up being there's a membrane in between these two ions that have positive and negative charges. Let's just say the negative ions are inside of the cell. Then you've got a membrane separating the positive ions sitting outside the cell. So there needs to be some sort of barrier, energetic barrier between them. And the measure of the potential energy generated by these separate charges. So the measure of the potential energy between this positive and negative ion is measured in something called volts. Within the body, it's on a very small scale, so we measure in millivolts. Voltage is always measured between 2 points, and it's called the potential difference, or simply the potential. And generally speaking, the greater the difference in the charge between 2 points, the higher the voltage. So the greater the difference between their positive and negative charges, the greater the voltage. The flow of the electrical charge from one point to another is called the current. The amount of charge that moves between 2 points the amount of charge that moves between 2 points depends on both the voltage as well as the resistance. What the resistance is basically the resistance or hindrance for that charge to flow, and voltage is the free-flowing measure. So substances with high electrical resistance are called insulators. Those with low resistance are called conductors. So how much you resist the charge reflects how well the charge can flow. So this is basically what we're working towards here. What we're looking at is something called the membrane potential. So we kind of just talked about this. We've got a negative environment and a positive environment. They are basically separated by this plasma membrane. So we're talking inside the cell with the negative charge, outside the cell with the positive. So the membrane potential is the difference between the positive and negative ions between the inside and outside of a cell. And there needs to be a barrier that keeps them apart. This is going to be some sort of plasma membrane. And when cells are in their resting state, they're considered polarized. So polar opposites is one way you could think about that. Typically speaking, a resting membrane potential, so the value of between the inside and the outside of a cell at resting state is a value of negative 70 millivolts. So remember that, that's important. Negative 70 millivolts is a resting membrane potential. The resting membrane potential is essentially a charge difference across the plasma membrane when the cell is in an unstimulated condition. The inside of the cell will be negatively charged compared to the outside of the cell. And you can see that here. So they're looking at basically within the axon of a nerve cell. And then you can see that the inside of that axon is negatively charged and right outside of it is positively charged at a value of negative 70 millivolts. So depolarization is a process. We've talked about depolarization, so now we're getting real definitionally specific about it. Depolarization is the decrease in the resting membrane potential. So the inside of the cell becomes less negative, or another way to say that is more positive. So if you look at this along the y-axis, you can see the millivolts of the membrane potential. And you can see that negative 70 resting membrane potential. And then basically, some of those negative ions inside the cell got exchanged outside, and some of those positive ions outside the cell got exchanged inside. So you change the relative polarity of the cell. So when that happens, you begin to depolarize because you are basically switching out those substances, and it's changing the relative charge between the inside and outside of the cell. So you can even see here, if you look at the number values, you're depolarizing more towards 0. So you're becoming less negative, more positive. So that's depolarization. Hyperpolarization is the opposite. So this is an increase in the resting membrane potential. So this is generally the cell becoming more negative. So after a depolarization, you need to hyperpolarize or repolarize. There's different ways to say it. You repolarize the cell by hyperpolarizing it. So what you do is you would start the changes you made from the depolarization when you started exchanging some of the cells inside for outside and changing the charge. You basically would reverse that process, put what was originally inside back inside, what was originally outside back outside, and you see a return to the normal resting membrane potential, or the way it actually works out in anatomy and physiology within our bodies is that initially we go past. So this is resting, negative 70. We go past that almost to negative 90. So you overbalance, and then the body balances itself back out to normal resting position. So it's all about the back and forth between depolarization and hyperpolarization and the conditions that cause those things to happen. So what causes those things to happen is basically the changing of the relative concentration of different chemicals inside and outside of the cell. So what we've got here is something called concentration differences across the membrane. So there's a higher concentration of K plus potassium ions inside the cell than outside, and a higher concentration of Na plus sodium ions outside the cell than inside. So you've got basically sodium outside the cell. You've got potassium inside the cell. And the whole idea of it is this is the resting ideal state. So when you start to depolarize, what you basically do is you start exchanging these chemicals. You grab 3 sodium and you eject them inside the cell. And then you take 2 potassium and you eject them outside the cell. And this starts changing the relative polarity between the inside and outside of the cell because you're taking these charged ions and you're changing their configurations. So basically, the whole idea is that the sodium-potassium pump depolarizes, repolarizes, or hyperpolarizes, and maintains resting membrane potential all through the interchange of sodium and potassium in varying configurations between the inside and outside of the cell. Let me see what's relevant on here. So basically, there is a little bit of leakage that happens naturally. So the cell always has to kind of balance this out between the potassium and the sodium, compensating with actively pumping the ions, because through the leaking, you will see too negative of a value. So then you got to balance it out to the -70, and you got to keep balancing basically. So we're going to go ahead and stop this video here, and we're going to pick it up on video 2 for the sake of spacing.